Good morning and welcome to Global Healthcast, brought to you by Global Health Press. In this broadcast series, once a week, we bring to you news about infectious diseases, vaccines, and vaccination. I am Joe Schmitt, and with me is Dr. Melvin Sanicas. Good morning, Melvin.
Good morning, Professor Schmidt, and good morning, good afternoon, or good evening to those who are watching us wherever you may be.
The topics we are covering today, as always, is the COVID-19 update, the update on monkeypox, some new events there, RSV vaccine efficacy in adults, first data available. Melvin will cover Crimean-Congo hemorrhagic fever. I will cover a personal success story on aerosolized amphotericin B for COVID-associated pulmonary aspergillosis. And finally, the good news is there is a second malaria vaccine approved. And Melvin, we start with COVID-19 update. What's the story?
Yes. So for those watching us for the past 33 episodes, you know that we start with an epi update of COVID, and we get this from the WHO. This is a situation report that they publish every week, and this is a week late, so these are not real-time reporting of COVID, but you will see that in the past 28 days, 3 million new cases and over 23,000 deaths were reported to the WHO. It seems like a lot of number, but that is actually a decrease of 28% in terms of cases and a decrease of 30% in terms of deaths compared to the previous 28 days. And that is a good sign. You will see here that the cases and the deaths are going down globally.
Interesting. I think just to remind people, this bar here is probably China, most cases from China, and there the epidemic is really waning. Numbers are substantially lower. And the question then, which countries are left with the disease, Melvin?
Yes. So here you will notice that quite a few, actually several countries are showing up red as compared to the previous global health cast. But this does not mean that all these countries actually are are in a bad situation. It's just that the cases there are increasing. So contrary to the overall trend of decrease in cases, we will see a lot of increases in cases and deaths in the Southeast Asia and the Eastern Mediterranean regions of the WHO. And this is primarily in countries like India and Nepal, where cases are increasing.
Interesting. So that is case numbers. And how does it look with regard to deaths?
Yeah, so in terms of death, you will also see that India is also part of the top in terms of mortalities, and this has been attributed to the variant that has been designated as a VUM, or a variant under monitoring by the WHO end of March. This is the XBB 1.16, which is related to the XBB 1.5, and it has a name actually. It's called Arcturus. And yeah, I mean, unlike the other XBBs, right? We just say numbers, XBB 1.5, but this one has a name. I don't know why. This subvariant has been detected in at least 29 countries so far. And yes, these are the countries in the Southern Asia and Eastern Mediterranean region of the WHO, where you will see all these red on the map.
Mhm. Melvin, we now covered case numbers, we covered deaths, and I guess the next slide is on hospitalizations and ICU admissions.
That's right. That's right. So, well, here the message is still similar to what we've mentioned in the past Global Health Cast, that the hospitalizations in general and the ICU admissions are down basically. Either they're staying down or they're going down. If you look at the graph on top, it's really it has gone down, and the January peak is because of the China situation that they had this outbreak in December and January.
Very interesting. So overall, good news. The increase of cases with this new variant doesn't show up yet here in the statistics. So
Yes, not yet.
not yet, yeah. Melvin, thank you for the update on COVID. And the second pandemic out there is the monkeypox. And here you have an update as well.
Yeah, for monkeypox, if you watch us, you would notice that we don't talk about this every week because we don't really have an update every week, right? Because what happened is at the global level, really the monkeypox or the MPOX cases continue to decline, except for two regions that have a that had a bit of an increase, and one is the Western Pacific region where there was an outbreak in Japan with some local transmission, and you will see the epi curve here. And then one is in France, where there have been 17 cases among people who've actually received 2 doses of the vaccine. And you will probably ask, so then what happened to them? They're all okay. They were infected, but because they were vaccinated, the disease was not as bad as the unvaccinated.
Yeah, that is interesting. And I think we should follow up on this. Right now, I guess we don't have enough information. And I think this is very important to know all about it.
Yep. We will probably read a publication on that specifically.
Yeah. Very good. Thank you very much for the two updates on monkeypox and on COVID-19. I cover a story today on one of the most common infectious diseases of mankind. Everybody has had RSV, respiratory syncytial virus infection. Once you are 2 years old, 50% even have had it twice. And even throughout your life, you will have repeated RSV infection because protective immunity does not exist. You can get it again and again and again. And I'll talk about that again in a minute. But the story is some years ago, the NIH researchers found why vaccines against RSV do not work well. And they found that the protective antigen, the antigen that it uses protective immunity, comes in two conformations, pre-fusion and post-fusion. And all vaccine efforts from the 1960s on, they were post-fusion F vaccines, and they never worked. And now with the identification of the pre-F protein and how to stabilize it, tons of companies are developing RSV vaccines. Because again, this is probably the most common infectious disease of mankind. And here you see the different platforms that have been developed. And you see here phase 3 studies, different platforms are producing or are right now in phase 3 with such vaccines. And on top here is RSV pre-F proteins. 2 products are in development. And last week, the New England Journal of Medicine had results from one of these trials. But before I want to go there, I want to convince you that there are a lot of trials ongoing and they have nice names. This is why I show the slide. The RSV trial by GlaxoSmithKline in adults 60 years and older, or the Renoir trial from Pfizer, that is what I will talk about today, the Evergreen study from Johnson & Johnson, and the Primrose study, again from Johnson & Johnson. I like these names and the innovations that comes with language. Isn't that fun? But what you would certainly like to know is, does the vaccine work? And here is the result. Published in the New England Journal of Medicine. This is actually an ongoing phase 3 trials in adults 60 years and older, 1-to-1 randomization, 1 dose of 120 microgram of RSV A and B subtype, 60 micrograms of each of the antigens. And the endpoints were efficacy against RSV-associated lower respiratory tract illness with either 2 or with 3 signs or symptoms or any respiratory tract illness. So this is the endpoint. And what you see here on top is, on the top left side, lower respiratory illness with at least 2 signs or symptoms. And again, this is a little bit misleading. The 96% is the confidence interval. This is the efficacy. It is 66%. Actually, in the trial, there were 34,000 participants, which is quite a large number. So 17,000 received the vaccine and 17,000 received the placebo. And again, this is a very good efficacy for a mild form. If you go for low respiratory illness with at least 3 signs or symptoms, efficacy goes up to 85.7%. If you go down again, and if you say RSV-associated acute respiratory illness, which is very unspecific, many cases don't even have RSV, right? No, they all have confirmed RSV, but it's very unspecific. Some runny nose, whatever you have, efficacy goes down to 62%. The story to be learned here is the concept that vaccines, like respiratory diseases, don't protect well for future illnesses. All these subjects, 60 plus, have had repeated infections with RSV in the past, and they were not protected. Now you had repeated infections in the past, you get one, let me call it booster dose, and efficacy of the vaccine varies between 62% for mild disease and 85% for more severe disease, which is not much. With measles, we are 99.99% with 2 doses. With the pneumococcal conjugate vaccines, we are in the 90%. But if you go to influenza, vaccine efficacy or effectiveness is in the 50% range and even lower. So the story learned is with acute respiratory pathogens, we are not good with reducing carriage and acquisition. So we again and again throughout our life get the disease. And the only thing vaccines can do is they can give you protection against severe disease and death. This is the story behind this that I wanted to communicate today. And I think this is all we have today. And maybe the research in the future should aim at developing vaccines that reduce carriage. Melvin, any questions on this, or shall we go to the next topic?
No, I think just to again emphasize to people that the 96% is not the confidence interval, is not the efficacy, but the confidence interval. And to be fair, these numbers, 67%, 86%, are actually good for a respiratory virus. Yeah. As you mentioned, if you compare it to the efficacy of influenza, for instance, this is much better. But if you compare it to the pneumo vaccine, this is way lower. So I mean, the other message would be, I guess, to look at the efficacy of vaccines relative to the diseases. It doesn't mean that if it's the same respiratory disease, then the efficacy would be the same as well, right? You know, it depends on the disease, and it also depends on the population, because as you mentioned, these are elderly individuals who have been infected with RSVs several times already in their lives.
Yeah. Interesting disease, interesting concepts. And let's move on to Crimean-Congo hemorrhagic fever.
Yes. So last week for GHC33, we discussed the Nipah or the Nipah virus. And what we're trying to do here is just really talk about the infectious diseases that the WHO think are really important in research. And it's not saying that others are not, but these are the ones that need to be prioritized because of the fact that there are no drugs or vaccines against these potential outbreaks. And today we will be covering the Crimean-Congo hemorrhagic fever, or the CCHF. So this is it's nicknamed the nosebleed fever because people who are infected with this CCHF usually present with nosebleed. They have been around for a long time, but it's now spreading on multiple continents, including throughout Europe. It is carried and transmitted most frequently by ticks of the genus Hyaloma, and the mortality rate is as high as 40%. 30 to 40%. And as I've mentioned, there is currently no known specific vaccine or treatment for this. And cases have been documented in many parts of the world, including Africa, Asia, the Middle East, Russia, and the Balkans. And recent and more frequent outbreaks have occurred in Western Europe, leading the WHO to really categorize CCHF as a priority pathogen.
Interesting. Yeah. And so the endemicity of the disease of the virus depends on the availability or of the presence of the respective tick, right? This tick has to be there if you want to, if there is to be any disease risk.
Yes, that's right.
Okay.
That's right.
All right.
And here, it's really just showing us the transmission of the virus, while this hemorrhagic fever, CCHF, is only carried and transmitted by ticks. There is actually a diversity in the vectors and the targets of other Bunya viruses, which this is actually a part of. Majority are vector-borne, and excluding the hantavirus and the arenavirus, all other viruses in the Bunya viralis order are transmitted by arthropods such as ticks, mosquitoes, midgets, and sandflies. And due to their segmented genome, Bunya viruses are capable of swift recombination, increasing the risk of outbreaks. And it is possible for the Crimean-Congo hemorrhagic fever, specifically this virus, to undergo recombination through the reassortment of genome segments as well as homologous recombination.
Yeah. Okay.
Which makes it
So
really interesting, and the potential for outbreak is really there, right? It's really evident.
yeah. So it's a complicated life cycle of the tick. And in addition, the virus has to be present. And I guess the reservoir animal is rabbits, some birds.
Yeah, it can be there. It can be in rabbits, in birds, and in livestock.
Yeah. But humans are not basically the reservoir, except for the rare case where the tick bites you while you have viremia.
Yeah, and while you are spending more and more time with it with these animals.
Yeah. Very good. Very interesting.
And I
And
think
here
this
are
is the
the,
last slide. Basically, again, to remind people that there is no vaccine or treatment for this. But this is a type of a hemorrhagic fever, which means it interferes with the body's ability to clot blood, right? Early symptoms are fairly general, like fever, muscle aches, headaches, and dizziness. Patients can also have nausea, diarrhea, and vomiting. And after a few days, as the disease progresses, there is severe bruising and nosebleeds, as well as a rapid heart rate and PTK, and a rash caused by bleeding into the skin. And severely ill patients can actually experience kidney, liver, or pulmonary failure after the fifth or sixth day of illness. And fatalities mostly occur in the second week after the onset of symptoms.
yeah. So basically, many of these dangerous diseases or viruses cause hemorrhagic fever. So it's an unspecific finding. They activate the clotting system, and then you have hemorrhages all over your body, blood clotting, basically.
That's
And
right.
then you have a high mortality, right? This is the basic story, right? How these all work. Yeah.
Yep.
Thank you, Melvin, for this update. Now we have the Nipah virus, and we have the Crim-Congo hemorrhagic fever. And next week, we will have another of the 9 most relevant infectious diseases where we have no treatment, but we're seeking to develop vaccines. I have now a very personal story, and the story is from a publication that I actually just saw this morning when I opened my computer. It was about the prophylactic nebulized liposomal amphotericin B2 use, the risk of COVID-associated pulmonary aspergillosis in mechanically ventilated COVID-19 patients in an ICU. Now, here is the story. I became a professor based on my research on pulmonary aspergillosis. Aspergillus is a fungus And spores of this fungus are in the air everywhere around the globe. Everybody inhales aspergillospores every day, and 7 of these spores make it down into the alveoli of your lungs. The treatment for aspergillosis, well, nobody gets sick with this, right? Your immune system takes care of that right away, nothing happens. But if you have an underlying disease like neutropenia, or if for any reason your immune system does not work well, then you may come down with disease with aspergillosis in the lungs. And now COVID infection is a risk factor for developing pulmonary aspergillosis. And again, this is called CAPA. There are treatments for aspergillosis, but they don't work well. They don't work well because it takes a long time before these drugs make it into the lungs. There are many new drugs available now, but again, the disadvantage is always before you find a trace of these drugs in the lung, takes some days. Now, for amphotericin B, I did this many, many years ago, and I found it takes a week before you find amphotericin B in your lungs, and you have to have daily treatments. At the same time, amphotericin B is highly toxic. Actually, my patients called it amphoterrible. You get fevers and chills with the infusion of amphotericin B. So this is not a nice drug to be treated with, but it is life-saving if you get it. Now, we had the idea in the 1980s, if you cannot tolerate it as an IV infusion, we inhale it. So we developed an animal model, and in the animal model, aerosolized amphotericin B was highly effective in preventing pulmonary aspergillosis. You inhale the drug, and it is in the lungs right away as of day 1. However, used as a treatment, it is, let's say, 50% effective. And if you use it as a prophylactic is 100% effective. So the story is you have to use it early on. Now, my personal success of the day is researchers in Nottingham did a retrospective analysis of their data between December 2020 and February 2021. So let's say 14 months, they had 12 patients with probable COVID-associated pulmonary aspergillosis. Then they started aerosolized amphotericin prophylaxis, and they only had 1 probable case. Okay, this is not a double-blind randomized study, right? There may be various reasons why case reduction was 90%, right? But it is really a clue that this may work. And the nice thing is, I have the patent for it, right? This is my patent from 20 years ago. It probably expired. It has no commercial value, and I would never claim anything on this, but I think it is a late success in my life that something I developed 30 years ago has now been used, and it may show signs of clinical efficacy. In the past, this type of prophylaxis for cancer patients was not much used, because it is associated with bronchospasms. But now it may be, with a liposomal amphotericin B, it may be better tolerated, and it may offer an opportunity to patients around the globe with CAPA. So, Melvin, this is my story of the day. I have to tell my wife, I have to tell the world, this is great news, my personal success story of the day.
Congratulations, Professor. I think it's really good that you saw this study, because I also relate this to the previous topic about RSV, right? I think people now have changed their mindset on how drugs and vaccines can be delivered. Maybe you can give your drug IV or IM, and maybe also intranasally as well together, and maybe the effect is different. And you've seen this now in other studies in COVID vaccines. In China, they've started this. So they have intramuscular vaccines, and then the booster is intranasal. So who knows? Maybe in the future, this will become more of a norm.
Yeah. Let's be innovative. And talking about innovations, you have another good piece of news for today for people in countries with malaria.
So there is some good news about malaria for endemic countries. The University of Oxford announced that Ghana has become the first country in the world to approve their malaria vaccine. It's the R21 Matrix M vaccine. It's licensed and made by the Serum Institute of India, however, developed by the University of Oxford. This is different from Mosquirix, right? The vaccine developed by GSK, which was approved a few years ago. And this is basically the second vaccine for malaria that's approved in Ghana. And unlike Mosquirix that's been approved in more countries, this is just a first start in Ghana, but it's a good start, right? Because Ghana is one of the countries that is really affected by malaria, and we will probably see the results of how effective really this malaria vaccine is going to be when used in the real world.
Very good. So we finished with really good news today from RSV, COVID and COVID numbers going down, RSV vaccine efficacy. We had liposomal amphotericin. We had vaccine development for Crimean-Congo hemorrhagic fever, and now we have a new malaria vaccine. This was a very innovative podcast today. And maybe I can summarize. It was here the topics that we covered. And Melvin, you have a last funny of the day, a last word.
It's not really funny today, right? It's just something I saw on the internet yesterday, and I thought it's really good to share because it's really true. All of us, all our opinions, whether scientific or non-scientific, it's loaded with personal history and personal experiences, right? We can never say that our opinion is bias or personal history free. So it's just something to think about that when we give judgment or we opine on something, it actually also tells the people about our own personal biases and history.
That is a nice piece that relates to artificial intelligence. Artificial intelligence has the problem that it has no consciousness, or maybe it's not a problem, maybe it's good it has no consciousness. Our consciousness is based on our experience, and this is why whatever we say and think, this is based on our past. And artificial intelligence does not have a past, right? It is what you tell the system to react on. Melvin, I think this was fun today. Thank you very much for joining us. Thank you, everybody, for listening. Please look at the show notes if you like, and give us a like or a star for our podcast today. And Melvin, I say goodbye to you and to our to the people listening to us today.
Yes, thank you very much. Stay safe. And I actually noticed that some of our past videos have increased some views. So I'm happy that you guys are watching the previous videos as well.
Yeah.
Thank you.
Bye-bye.